Hi everyone, it's Mrs. Moore here, ready to take your Chapter 9 bio test. Now, I'm going to read through each question, but if I'm going too fast, make sure you pause this video and go at your own pace. Okay, here we go. Make sure you're filling in your first name, last name, and your class period. And once you're ready, we're going to start with number 1. Genetic engineering is possible because all organisms are based on the same proteins, DNA copies, plasmid loops, or genetic code. Scientists can study the effect of turning off a gene by using cloned animals, gene knockout organisms, DNA microarrays, or DNA fingerprinting. Number 3. What is a clone? A genetically identical copy of a gene or organism, a ring of bacterial DNA with foreign DNA inserted, a mouse that has some of its genes turned off, or a genetically engineered organism with new traits. Number 4. Which of the following technologies uses electricity to sort DNA fragments based on their sizes? Genetic screening, gel electrophoresis, DNA microarrays, or genetic engineering? Here is a figure 9.1. You're going to use this for the next question, so make sure you refer back to it. Number 5. In the restriction map shown in figure 9.1, which line or band represents the smallest DNA fragments? Make sure you're using that picture. Is it A, B, C, or D? Let's go to the next section, section 4. Number 6. Which of the following processes could a scientist use to make an amount of DNA large enough to study? Polymerase chain reaction, or PCR, genetic engineering, bioinformatics, or gel electrophoresis? Number 7. Which of the following is the study of entire genomes? Gene sequencing, proteomics, genomics, or transgenetics? Number 8. In polymerase chain reaction, which of the following acts as the starting point for new DNA strands? cDNA, plasmids, enzymes, or primers? Number 9. The restriction enzymes used by scientists to cut DNA molecules at specific nucleotide sequences come from mice, yeasts, bacteria, or viruses? Number 10. Which of the following experimental techniques involves treating a genetic disorder by replacing a defective gene with a functional gene? Genetic screening, bioinformatics, genomics, or gene therapy? Number 11. Suppose the restriction site for a particular restriction enzyme is GATC on the top strand and CTAG on the bottom strand. The enzyme cuts the DNA between the A and the T on the top strand and between the T and the A on the bottom strand. How many restriction sites for this enzyme are there in the DNA sequence shown in figure 9.2? Using that information and this picture right here, answer this question. Is it 2, 3, 4, or 5? Number 12. What goal of the Human Genome Project has been achieved? Learning how DNA sequences vary? Sequencing all the base pairs of human chromosomes? Understanding the function of each human gene? Or discovering the proteins that play a part in cancer? All right, number 13. Genetic screening can help determine a person's risk of passing on a genetic disorder through a combination of family history and recombinant DNA, gene knockouts, DNA microarrays, or DNA testing. Number 14. The purpose of genetic engineering is to develop safer pesticides, study obesity and cancer, reduce Earth's biodiversity, or give organisms new traits. When you're done with that section, let's keep going to section number 5. Number 15. Proteomics is a study and comparison of proteins within and among species, genomes of various organisms, DNA sequence variations among people, or transgenic organisms in different environments. All right, here is figure 9.3. Make sure you take a really good look at that because you're going to be using it for the next question. Number 16 says, what is shown in figure 9.3? Recombinant DNA, a series of nucleotide sequences, DNA fingerprints for 3 people, or transgenic organisms. Number 17 also uses figure 9.3, so make sure you look back at the picture to answer the question. Number 17 says, what is this type of DNA, or figure 9.3, used for? To identify people at the molecular level, study human migration patterns and genealogy, study biodiversity, or all of the above. Number 18 also uses figure 9.3, so make sure you look back at it to answer the question. Number 18 says, what does each of the bands in the diagram represent? The number of repeating nucleotide sequences, or the size of DNA fragments which depend on the number of repeating nucleotide sequences? Number 19. The analysis of DNA shown in figure 9.3 comes from a mother, a father, and their child. Could person 3 be their child? Yes, person 3 could be the child of person 1 and 2. Or no, person 3 could not be the child of person 1 and 2. Again, make sure you're looking back in figure 9.3 to help you answer the question. All right. Now you will see figure 9.4. Make sure you're using this picture to answer the following questions. Number 20 says, identify the structure in part 1 of figure 9.4. recombinant DNA, a cap, plasmid, or tail? That is structure 1. Use that picture. Number 21 says, where is the structure in part 1 of figure 9.4 found? Again, look back at that picture above. Is it in the bacteria, yeasts, or eukaryotes? Again, for number 22, you will use figure 9.4. 22 says, what role do restriction enzymes play in part 2 of figure 9.4? Restriction enzymes insert new DNA into bacterial plasmids. Restriction enzymes cut plasmid DNA and foreign DNA to leave complementary sticky ends. Number 23 also uses figure 9.4. Number 23 says, what is the term for the structure in part 3 of figure 9.4? Recombinant DNA, transgenic, gene knockout, or clone? Number 24 says, an organism that has one or more genes from another organism inserted into its genome is said to be recombinant, mutant, transgenic, or bigenetic. And your last question, number 25. A scientist wants to insert a gene into a plasmid as shown in figure 9.4. In order to open the plasmid to insert the gene, the scientist must use restriction mapping, PCR, restriction sites, or restriction enzymes. That is your whole test read aloud to you. Make sure if you need a question repeated, you are going back and listening to it as many times as you need. Good luck!